out now.
I'll go turn it up too.
Sweet, thanks.
Yep.
Hey everyone, I'm Bryce. I'm a postdoc at University of Washington. I work in Maitreya Dunham's lab in the Genome Sciences department, and today I'm going to share with you a course-based research experience I've been developing. So my background is in yeast genetics and evolution, and I really like working with yeast for a couple reasons. One is that yeast is a really awesome model organism for genetics because we have this really robust toolkit. We can do things like genetic engineering. It's very easy to sequence their genome. And these techniques allow us to do things like sort of peek under the hood at evolution and really see how adaptation works. So let's say that, like me, you're a fan of foreshadowing and you wanted to evolve yeast to deal with a drug like clotrimazole. You can, over the course of several weeks of selecting for yeast that can resist this drug, you can have hundreds of generations occur and select for mutants. And then using that toolkit, you can peek under the hood, the molecular biology, and see the mechanisms by which yeast becomes resistant to the drug. And this leads into the second thing that I really like about yeast, is that they're a really nice organism for teaching. It doesn't take a whole lot of resources to get a yeast lab up and running. And because of the incredible molecular biology tools and that ability to see what's happening in evolution, students can really see what's happening at many different levels and hopefully get a good vision of how evolution works. So I believe that as a scientist, it's part of my responsibility to engage with the public. And every time I do this, I feel like there's this really nice feedback where I try to share my science with people. And through that, I get this feedback where I learn how to contextualize my research and learn how to better engage the public. But as a researcher, there are a lot of different pushes and pulls on my time, and it can make it hard to invest the time to both develop and then carry out and maintain long-term collaborations in outreach. And so I've been using yeast as a model organism for teaching so that I can develop projects where if I work with teachers to develop research exercises for students, we can give students an authentic research experience. And if we design things the right way, those students can then generate data that can feed back into the ongoing research projects in my lab. So one way I've gone about this is developing something called yEvo with a teacher named Ryan Scopehammer at the Westridge School in Pasadena, California. This is a private girls' school, and Ryan, during his PhD, became interested in evolution and was inspired by work from Rich Linsky's lab on long-term evolution in E. coli. And wanted to bring some sort of evolution project to his high school classroom. The AP Biology curriculum is centered around eukaryotes, so we wanted to use a eukaryotic model for this. And so we developed what we're calling yEvo, which is a very creative combination of the words yeast and evolution. Going into it, we had some basic design goals, like we wanted to give the students an authentic research experience. We wanted to have opportunities for both collaboration and friendly competition. And we wanted to give students a full view of how evolution works at the molecular level. So one question that Ryan and his students were interested in was the rise of antifungal resistance globally. There are a lot of different antifungals that we use to treat fungal infections, and pathogenic yeasts are rapidly becoming resistant to these. And Saccharomyces cerevisiae, my favorite yeast, is a nice domesticated model for studying this phenomenon. Even though it's been domesticated, it's something that we literally eat and drink all the time. The genetic pathways underlying resistance to many of these antifungals are conserved. And so if you, there we go. So clotrimazole is a drug in this compound called Fungicure. When it enters the cell, it inhibits an enzyme called ERG11 that functions in ergosterol biosynthesis. This is basically the yeast equivalent of cholesterol. That pathway is actually conserved from yeast to humans just about. That drug can be exported through these very conveniently named multi-drug efflux pumps, and those are regulated by transcription factors called PDR1 and PDR3. So there's a very nice mechanism for this. There's a fair bit known about how this works. but we thought that doing a long-term evolution experiment using this compound could potentially provide some new information about mechanisms of resistance. So we set out to design an evolution protocol to study this. I'm not going to have time to go into all the details about how we designed it and what we found, but I'm going to try to give you a vertical slice of that process today. We instituted several measures to ensure that students are culturing only their yeast and nothing else, as well as a bunch of additional measures to make sure that students are getting the most out of this experience that they could. So the way the protocol worked, every day students would come into class, they would grab their test tube of yeast and make some observations about it. They would record how well the yeast was growing in the presence of the fungicure, and then they would make a decision about whether they wanted to up the dose or keep it the same. They would then add some fungicure to a fresh tube of media, and then use sterile swabs to take a little bit of their yeast culture and put it into a new tube. So one thing I haven't told you about the yeast yet is that we didn't just use any old lab strain. We actually acquired some engineered colorful yeasts from our collaborators at the Jeff Buka lab at New York University. These yeasts are carrying a plasmid that provides them with resistance to a second antibiotic that keeps lowers contamination risks in their cultures. And the colors provide a useful marker for competition experiments. So each student group chooses their favorite color from this living palette. And after they've evolved their yeast, they can mix two different colors together, compete them in the presence of fungicure, and then plate them and count colony-forming units of each color to determine which one was more resistant. So Ryan decided to set this up as a bracket. So at the end of the experiment, they carried out a yeast madness. The students were not as into March Madness and decided to name all their yeast after Pokemon. I think last year Snorlax won because, of course, I mean, it's an electric tape, it's not going to work out. And the students got really into this aspect of the experiment. Ryan's told some really funny stories about students like coming into class early to read each other's notebooks so they can figure out if like I'm team blue and team black has got like way more fungicure, I gotta go crazy and up my dose today. So the original plan was for them to evolve their yeast for 7 weeks, but because they wanted to win this competition and because Ryan got really into the process, they ended up keeping the experiment going throughout the school year, and this became something that just was part of their class culture and part of their routine whenever they came to class. So at a few different time points during the experiment, Ryan sent up some glycerol stocks of the students' populations. I struck these out onto agar media to isolate clones. I then phenotyped these and chose the most resistant clone from each student experiment. I sequenced them, curated the mutation data that I found, and then sent that back to the students so that they could start looking at their mutations from their yeast. So there are a bunch of really nice free tools that are available in the genomics community and in the yeast community in particular. One is the integrative genomics viewer, which lets you look at mutation data and see the changes to sequence and what gene they fall into. And then another one called the Saccharomyces Genome Database is a repository of all the data from yeast experiments over the last several decades, and has very nice curated lists of papers published on each gene so that students could be guided through a literature search to figure out what their mutations might be doing at the molecular level. So to give you a taste of some of the data, this is from a genotype from group G. This is a list of genes or types of mutations that are very common in the literature that contribute to azole resistance. And group G had mutations in 2 genes called PDR3 and ERG11. So I don't want to go too deep into the molecular biology, but ERG11 is that enzyme that's inhibited by clotrimazole. And this mutation led to 2 copies of that gene being present in these yeasts, so they can now deal with that inhibitory effect a little bit better. And PDR3 is the transcription factor that leads to expression of those really convenient pumps. And it's been shown in pathogenic species of yeast that gain of function mutations in this transcription factor lead to increased expression of the pump. So it's now easier to get stuff out of the cell. And the transcription factor result in particular was something that Ryan was really excited about. He loves gene expression. And so he tied this into his class's discussions of gene expression and gene regulation during their class. So to zoom out and look at all the data that we've collected so far, this is a bunch of different clones that we've sequenced. And one thing that I'd like to highlight here is that every single student clone that we sequenced had at least one mutation with a very clear role in azole resistance. And at this point where we started sending back the data to the students, this is where the collaboration came in, because they were each looking at their yeast and their mutations, but by working together and looking at what each other group got, they can find these patterns where they're seeing things recurring many times throughout the experiments. And this isn't all the data that we have, because in addition to these well-characterized genes, there are mutations in over 120 additional genes. And because of the length of the experiment and the number of replicates we have, I've started looking through to see if there are any patterns in occurrence, and I can't go into it yet, but I think I might see this one instance of epistasis where there are two mutations that always co-occur. And so this summer I'm working with an undergraduate in our lab, Angela Hickey, to follow up on this and see if that's actually what we're seeing. So the next thing we were curious about is what impact participation in this has on student learning. So we partnered with Alexa Warwick at Michigan State University to design a set of surveys that we give to students before and after the experiment. In year one, we just gave a post-lab survey, and that helped us to identify things that we could improve going forward. One of my favorite quotes from that survey was in regards to the sequence analysis. Very dizzying, lots of letters, which I totally agree with. I mean, that's definitely true. So we made some changes to the sequencing analysis for year two, and are now analyzing some pre and post lab surveys and are performing interviews with the students. So as I mentioned at the beginning, Ryan works at a private school. He has a lot of flexibility in his curriculum, which is not the case at many schools. And so we wanted to see if we could implement this in a public school setting. And earlier this year, we partnered with yeast friend Paul Rowley and his lab, Angela Crabtree and Josie Boyer, who were both interested in outreach projects. And they took all of our protocols and strains and data and went to local Moscow high school to implement yEvo with their students. And we're now looking at some surveys from those students to see how the modified protocols that we implemented there worked out. So just to come back to this cycle, I feel like developing projects like this has been a really nice opportunity for us to get a really good feedback loop going in our research and outreach. And this is not something that is like a new idea. Cures do a really great job at this, and citizen science projects do this really well. And I've really enjoyed trying to implement this model with yeast evolution research. So I want to acknowledge everyone that's been involved in this project. I've already said a lot of names throughout this talk, but I particularly want to thank Maitreya Dunham, my PI. She's been incredibly supportive of me pursuing this project and is just a really all-around phenomenal mentor. And I also want to thank one of our funding agencies, Beacon. All of our funding agencies are great, but Beacon is particularly awesome because they really encourage inter-institution collaborations, and without them, I would not have met Alexa or Paul, who have been really instrumental in this. So with that, I'll take any questions.
Something like this would be, and I'm sure there's some out there, easily modifiable for undergrad.
Yeah, totally. Yeah, I think this would be.
That's got a different timeline, right? You make like once a week instead of every day.
Yeah, I think this would work awesome at an undergrad. I think it would be great at a community college, especially like a biotech program. There's a lot of cool applications of yeast to biotechnology and synthetic biology. I am currently entering the job market, so I'm hoping that wherever I end up, I can implement something like this as a cure.
Are there any, for someone who would want to implement lesson plans like this at an undergrad level, are there available resources for developing lesson plans and syllabi?
Yeah, there's some grants for that stuff, and I can talk to you about that after. For this particular project, we're writing up some of the lesson plans right now, but I'd be happy to talk to you about some of the stuff that we've learned from this. Hey, I think I should probably move on.